in my work because I agree with Joan Kaila that eradication is truly a dream. But I was charged in trying to discuss how new drugs or new treatment regimens for tuberculosis can get us closer to TB eradication. So you can start listening now, or you can stop listening now, because the answer at the end is going to be that new drugs is not the answer for eradication. But the vaccine definitely could be, and I hope very much that the fruity vaccine or some other vaccine gets us there. So the fact that it's still a dream and that the dream has been dreamt many times is well illustrated by Dr. Waxman when he received the Nobel Prize for medicine. He gave in his lecture, as you know, he's the discoverer of streptomycin. So when they discovered streptomycin, they thought it was the beginning of the realization of the dream. But in fact, quickly from the start, they discovered that, for example, you cannot use streptomycin by itself because it creates drug resistance. So the second drug that they combined with streptomycin was called PAS, a drug that we still use for the management of multi-drug resistant TB patients. And with that, they could control the fact that even after as soon as 2 to 4 weeks of streptomycin alone treatment, you get drug resistance. So in addition, in 1952, they discovered isoniazid. And with that, and when Dr. Waxman gave his acceptance lecture for the Nobel Prize, he said that the conquest of the Great White Plague, undreamt of less than 10 years ago, is now virtually within sight. So now you can calculate how many years since 1952 we're still answering the same question. To get a new drug is complicated, but even once you have the new drug, then is how do you use it in a new treatment regimen? Because a new drug you can never use by itself. In tuberculosis, the same way it happened with streptomycin at the beginning, you have to use it in combination with other drugs so you don't generate drug resistance. So this is just a little summary of this publication from 1999 in the International Union for Tuberculosis and Lung Diseases, which they did an overview of a lot of the clinical trials that had been published to date. And if you count them all, the number of trials that they had to conduct with how many different regimens they had to test, how many patient volunteers participated until they got to the short course 6 months treatment that we still use today. The trials for tuberculosis are extremely complicated because of many, many reasons, including that the trial's capacity, the groups that do the trials is limited. They are expensive, they are long in duration, because normally the endpoints that you're looking for are either the cure or absence of relapse when you finish treatment. So you have to treat the patients and then follow them for an amount of time to assure that they are not going to relapse. And that takes, between enrollment and follow-up, maybe 5 to 10 years to test maybe one drug. And if you're lucky to have more than one drug being tested in a drug regimen, it still takes that long. We're looking for surrogates, which would be something that you can measure way before you're measuring the endpoint of relapse, for example. It would make the trial much shorter. However, most of the the pharmaceutical companies that have the drugs, their main endpoint is also to get the drugs registered with European Medical Agency or with the FDA. And it would have to be that those regulatory agencies accept the surrogates so that you can do the trial measuring the surrogates and then use that data to go for registration, which is still, of course, difficult. The trials, once they get done, now it's getting better because there's standards in the way you code the data. It's called CDISC in particular for tuberculosis. But the thing is that even the CDC latest trials are not coded into CDISC, and when you finish your study, you still have to invest more money to transform all the data into the standards, because that's what the regulatory agencies will require. Nowadays, it's better to do the trial and have a good informatico and have them code the data from the start with the proper data standards. But there's also the lack of standardization of laboratory methods when you have to use multiple laboratories and not just one laboratory. The way you analyze the data is also sometimes different. And the better we get at making one unique type of analysis and results, then the better we will do in terms of making the trials faster. The drug, this may or may not be an issue, meaning that, again, the drug that is being tested is usually either sponsored or donated by the pharmaceutical company to the clinical trials group to get it tested. But the companion drugs that you have to use, be it rifampin or any other companion drug that you combine with the regimen, those usually, because of money concerns, they don't come from a central source. You have to buy them, source them from the source where the study is being conducted. And even though drug quality trials is supposed to be very well standardized, it doesn't always happen. And if you need a placebo, also has to be standardized, also very expensive, very difficult to produce. And unless the pharmaceutical company is willing to provide both the active drug and the placebo, usually the trial could become unaffordable. But we do have new drugs, and we are testing them in new drug trials. This is an illustration of the either older drugs that are being tested for different forms of administration in different regimens, or for, let's say, linezolid for multi-drug resistant tuberculosis, or rifampicin for drug-sensitive tuberculosis. But also the newest drugs that are still in phase 2 studies, maybe beginning phase 3 trials, that are really what we're going to analyze in terms of the question, can they get us to TB eradication? Well, this is more of the problems with how long did it take to get any new drugs to even be able to be here and talking about the new drugs. The drugs that are in use today, they were all developed between 1943 with streptomycin and 1963 with rifampin. And those are really the only TB drugs that are really effective and that we know very well how to use. The drugs in the 80s and in the 90s are really not new. They're new for tuberculosis, but they're not new for the world. They're the fluoroquinolones and the oxazolidinones, which is the linezolid family. And then, like I said, the rifamycins. But the new agents are really from this year's 2000s, and they're still not fully developed in the sense that we still don't know what role they will play in the regimens. The trials capacity, even though the BMRC used to be the ultimate trials group for tuberculosis, they of course are no longer actively conducting clinical trials. The union doesn't actively conduct clinical trials, but the Tuberculosis Trials Consortium in the United States since 1998 has been conducting clinical trials in tuberculosis. That's where I work in the clinical research branch, Division of Tuberculosis Elimination. We do our clinical trials in TB control programs, and so it's a little bit different than the pharmaceutical trials that some of you are familiar with. They don't happen in hospitalized patients. They don't happen with big pharmaceutical support. They are funded by the US government, and we have the data and coordinating center at the CDC. But we have a mission, which is to conduct studies that are programmatically relevant. So, meaning that the clinical trials must answer a public health question, not necessarily the most ultimate new drug, but the most important types of questions. And the types of questions that one could answer, perhaps we cannot eradicate, but perhaps we can make treatment better, or shorter in duration, or easier to administer, or less toxic. And so at the TBTC, our mission is not to find the new drug or to take the new drug to registration. That's for a company to do. In the TBTC, we focus in making the drugs or the drug regimen better. So we want more effective duration of treatment with less duration, with maybe easier to administer because the dosing is less frequent, that the patients have better results, less risk of relapse, better identification of the LTBI or latent tuberculosis infection type patients that actually need treatment, so you don't have to treat a lot of people that don't really need treatment. You treat just the few that will progress to active disease, and safer and better tolerated treatment. This is our clinical centers that we have across the world. We have one with Dr. Neil Martinson in Soweto, Johannesburg, in South Africa. We have one in Barcelona, and that's the reason they keep inviting me to come and give the talks here, because we've been collaborators for many years, and I'm happy to say it works very well. There is another big clinical trials group that didn't used to do tuberculosis trials per se, except when they pertain to HIV infected and in AIDS patients, which is the ACTG. From 2013 onward, the ACTG, their director, Dr. Fauci, has determined that the ACTG will work in the opportunistic infections of importance, and tuberculosis is the very, very top of the list, and they are no longer just going to do studies in HIV-infected patients. They are also going to do it in all types of patients, which will make a lot of difference, because the TBTC, relatively speaking, is a small group. We're funded for about $10 million per year, which sounds large in some contexts, but is very, very small in the context of clinical trials. The ACTG is, I don't know if it might be 10 times as big in budget. It is about 10 times as big in the number of clinical trial sites. This is a trial sites that exist right now. When they make the recompetition, which is to reestablish the sites in 2013 and 2014, they might not be the same ones that you see here, but they, as you know, they could be similar depending on who wins the new ACTG competitive process. So this is just to explain again that the studies that they normally do have to do with HIV and tuberculosis, but the fourth study there from the top, this, the one that says evaluation of gene expert in low incidence population, that one, for example, is the beginning of the new phase where they are doing studies in non-HIV infected population. That one, we are doing it in collaboration with the ACTG, because at least in the United States, the ACTG centers really do not see patients that, it would be very difficult for them to recruit tuberculosis patients without HIV infection. They don't get the care in the ACTG type centers, but the TBTC centers, they work in public health departments like they do TB care here in Spain. So we are going to be recruiting tuberculosis suspects to be testing the gene expert technology, which, as you know, is licensed to be used in Europe and in other places, but not in the US, at least not officially by the FDA, because the data in low incidence populations is not yet available. So we're going to do that study, and in fact, recruitment will begin next week. So the new clinical trials designs that have been approved to try to accelerate the process is the use of to test your drugs in multi-drug resistant tuberculosis. And the idea at the beginning, maybe less than 10 years ago, was that if you only did your studies in multi-drug resistant tuberculosis, you could not use them to get registration. You couldn't go to the EMEA or to the FDA. Little by little, the science logic of the fact that if you, all the companion drugs that you use with a new drug are usually very low bactericidal potency. So if you test a new regimen with what is called a companion regimen, meaning you use the new drug that you're testing with anything that's available and that the patient, whose strains the patient is susceptible to, that combination is called like new drug versus best available. That model of clinical drug testing is the way you do it in other diseases, but it's not the traditional way you do it in tuberculosis. But that model allows you to do it faster, because if a patient does better, it means that the new drug is doing a lot of efficacy, because the old drugs, the combination of that plus the fact that new drugs have to get moving quickly, the same as HIV drugs got moving quickly, then you have to change your ways. So this is the change. And for example, the TMC-207 was tested in an MDR-TB population, was published in the New England Journal. They did find that, for example, if you added TMC to the regimen, your conversion after 2 months of treatment was more than 3 times as high, and with very good statistical proof of that case. But will it become the answer to TB eradication? Well, the new drug doesn't just have to convert the sputum at the end of 2 months. It really has to make the patient bacterial load sterile, so that they don't get regrowth later. So for that, the TMC-207 and none of the new drugs have that type of follow-up that one can tell. You still have to go back to the mouse model and see how they do. But even in the mouse model, while the regimens that are listed here, where you see a J, because TMC, TMC used to be that's their acronym because of the Johnson & Johnson. So they're very good during the initial phase of healing, which is the induction phase of treatment. But after the end of treatment, you still have growth returning. So it means that you los marearon a los bacilos, you kind of like were going there, but because you don't have the right companion drugs, you don't completely kill them all. And the ones that are very slow growing, they're called persisters or slow growers in some other of the publications. I'm simplifying all of this because I didn't prepare the talk to talk about TMC-207. TMC-207 is used as an example of whether the new drugs can answer the question of TB eradication, which I think is what it's been discussed here. So TMC-207 will be a potent drug in the arsenal to treat tuberculosis. You can improve your intensive phase, and instead of having a 9% conversion, maybe you can get a 50% conversion on very difficult to treat patients with MDR-TB. But we still don't know very well what the companion drugs should be. For example, an old drug, pyrazinamide has been investigated at higher doses as a companion drug. Rifampin has interaction with TMC-207, so for drug susceptible TB, it's going to be difficult to use them together. TMC-207 is not a sterilizing agent, therefore it's not expected to be active against persisters. if we don't have accelerated path to regimens by all of us working together, the TBTC, the TB Alliance, the mouse modelers, they are normally at Johns Hopkins University, and now there's some new ones that moved to South Africa. St. George's University, where Professor Mitchison works in London. We're all after the same thing. We need new validated endpoints. We need to have maybe a different animal model, different than the mouse model. We need to analyze the data sooner and faster, and with trying to get the answer better. So in conclusion, what we do need is a stronger, shorter, safer regimen. But even if we, at this moment, could eliminate all the cases of tuberculosis disease, the large pool of people that are latently infected with tuberculosis would still serve as a significant reservoir for development of new tuberculosis cases for decades. So we need to continue to strengthen the infrastructure for clinical research, which brings the research study into the public health clinics and TB programs located across the world. Because the patients that participate in the clinical trials, the patient volunteers, get very good TB treatment. And in the case of MDR TB patients, they actually might get a curative treatment as opposed to no treatment at all or very deficient treatment. And then we need to continue effective and efficient mechanisms for protocol development and implementation of standardized instruments for the conduct of tuberculosis clinical trials. But in the last conclusion is that new drugs and new treatment regimens are unlikely to get us to tuberculosis eradication. Thank you.